Bismillahirrahmanirrahim. Assalamu alaikum. This is Miss Anam, microbiologist and MPhil scholar, and today's our topic of presentation is virus, basic characteristics about virus life cycles including lysogenic and lytic cycles, viral replication basic process, temperate phage, lysogeny, and lambda phase. So let's just start our topic, and before starting our topic, I request you all please subscribe my channel and press the bell icon. Virus is an infectious agent of small size and simple composition that can multiply in living cells of animal, plant or bacteria. In short, virus is a basic non-cellular particle which can't replicate outside the host. It's obligate intracellular parasite. Now coming to the next slide. Viral replication, the basic process of viral replication or viral infection. In first picture is shown that during replication process only viral genome injected into host cell, and in second picture during such replication process whole virus particle enters the host cell. These are the two basic pictures showing the viral replication process. Virus life cycles, the two reproductive cycles are lytic cycle and a lysogenic cycle. The lytic cycle consists of attachment, penetration, synthesis, assembly and release through lysis of the cell. And the other cycle, which is lysogenic cycle, the viral genome becomes integrated into the host genome and don't lyse or kill the cell. It is here shown that the lytic and lysogenic cycle, how these cycles are being conveyed. In the picture, first cycle is representing the lytic cycle and the other one is representing the lysogenic cycle. What happened in the lytic cycle? The viral genome is entered into the host and host cell replicate it and assemble the new viruses, and at the end the lysis of a cell is the destiny of this host cell, producing the viral genome. On the other hand, the lysogenic cycle, the virus integrate its genetic viral genome, and this viral genome integrated into the bacterial genome. Here the lysogenic cycle starts, and whenever the host cell replicates the viral genome replicates with the host genome, and at the stage when the cell is ready, the viral genome having host cell enters from lysogenic to lytic cycle and starts the lytic cycle, which is designed to lyse the cell, producing more viral genome having virus particles. The lysogenic cycle or the lysogeny. Lysogeny state is characterized by integration of the bacteriophage nucleic acid into the host bacterium's genome or formations of a circular replicon in the bacterial cytoplasm. Prophage. What is prophage? Prophage is the viral genome viral genome becomes integrated into the host genome. The viral genome is then called prophage. I can show you here on the previous picture. Here the viral genome integrated into the host genome, and this viral genome is represented here as a prophage. Now coming to the next slide, the lysogenic cycle, in which after penetration the viral genome becomes integrated into the host genome. A prophage gets replicated every time the host cell replicates and divides. A prophage can be induced in favorable conditions such as UV radiation or the presence of certain chemicals or other nutritional and environmental conditions, and can return to lytic cycle. A prophage, here is shown that how the prophage is being induced in favorable condition and can return to lytic cycle. And then coming to the next slide, the lysogenic cycle, here is completely represent the lysogenic cycle from step 1 to step last. From step 2, lytic cycle enters lysogenic cycle in unfavorable conditions. What happened in lysogenic cycle? Entry, viral DNA injected into host. Secondly, integration, viral DNA becomes part of host DNA. Here it becomes the part of host DNA. And fourth step, cell multiplication. Host replicates with viral DNA. The host replicating with the viral DNA and many cells divisions. At the last, cell enters back into lytic cycle and assemble and release and lysis of the host cell. Coming back to the lytic cycle. Now the steps of a lysogenic cycle are being explained very well here. First of all, viral genome enters cell. Viral genome integrates into host cell genome. Host cell DNA polymerase copies viral chromosomes. Cell divides and viral chromosomes are transmitted to cells, daughter cells, at any moment when the virus is triggered. The viral genome detaches from the host cell DNA and enters stage 2 of lytic cycle. Now coming to the next slide, temperate bacteriophages and lysogeny. Many DNA phages are temperate phages that have two options for replication: lysogeny or the lytic cycle. Advantages of lysogenic cycle: lysogenic bacteria have immunity to superinfection and under appropriate conditions they lyse and release phage particles. Lysogenic conversion: this occurs when a temperate phage induces a change in the phenotype of its host. Now the temperate phage, this is how the temperate phage chooses and divides the genetic material. Temperate phages have two reproductive options. In the first is replicate by the lytic cycle, and other option is the lysogenic cycle, in which occasionally a prophage may leave the bacterial chromosome and this phage DNA circularized and entered into the lytic cycle in favorable conditions, which I have explained earlier. Now the lambda phage, which is the example of a bacteriophage. Icosahedral double-stranded DNA phage that infects the K12 strain of E. coli. Here is shown the bacteriophage lambda. The cascade of events leading to either lysogeny or the lytic cycle involves number of regulatory proteins that function as repressors or activators or both. Two regulatory proteins are of particular importance: the lambda repressor product of the C1 gene, the CRO protein product of the CRO gene. The lambda repressor promotes lysogeny, prevents transcription of the viral genes. The CRO protein promotes the lytic cycle, and this is how the cascade of the genome of lambda phage happens, and how this gene is being activated the lysogenic cycle and enters the lysogenic cycle, or decides whether to enter the lytic cycle or the lysogenic cycle. Thank you. I hope my video will be helpful for you. And at the end, please I request you all to subscribe my channel. And Allah Hafiz. Take care.